Hi everyone. In this video, we will be discussing more about cell reproduction and cell division. So, when we think about the cell cycle itself, it has two major phases: the mitotic phase or the M phase, and a non-dividing phase called interphase. So this is how that would look with mitotic cells, where the DNA itself is on opposite edges. And then a cell that is in interphase is where the DNA itself is relaxed. Mitosis occurs very quickly, and once the cell begins, it completes, it takes less than an hour to kind of complete it. So now there are two types of cell division. One is meiosis, where it's the production of eggs or eggs and sperms, which are the gametes. These cells in the product have half the amount of DNA as the parent cell. This is for sexual reproduction, and we will talk about more about meiosis in the next video. So in this video, we are primarily going to focus on mitosis, where it is the production of body cells or somatic cells, where they are having daughter cells that are genetically identical to the parent cell, having two sets of chromosomes that are diploid, basically diploid. And the function of this is more in terms of like growth, repair, asexual reproduction, so on. And then both forms of these cell division uh, are accompanied by a process called cytokinesis, which is the division of cytoplasm into two distinct cells. Now, what exactly is a chromosome? A chromosome is a section of DNA and its associated proteins where a gene is a part of the section, part of the DNA where it encodes for specific RNA or protein. A genome is a complete set of it. And then chromosomes are basically a section of those genomes. So, one thing about eukaryotic DNA is that it is very, very long and it has to be condensed down into these compact forms of chromosomes to actually fit into the nucleus because it can wrap around the Earth a couple of times or the moon or go back from the moon and back a couple of times if you stretch it out. So we have to fit it in to that cell itself. So this is kind of the organization of eukaryotic chromosomes. It starts from the helix. The helix ends up wrapped around histone proteins, and then those become nucleosomes, boiled into like a fiber like this, and then it's further condensed into chromatin like this, and then that ends up with the duplicated chromosome itself. So now, one thing about chromosomes is that they can be stained with different dyes and have band pattern characteristics where they are distinct between different chromosomes. They can be arranged in the way of size, which basically tells you more about the karyosome. And the chromosomes themselves are organized by size in this case, where the largest is the first one and the smallest is number 22, which are the 22 autosome chromosomes. And then there's the two sex chromosomes, X and Y. Now, ploidy itself is a term that refers to the number of sets in a cell, where n is how many unique chromosomes make up a set. In humans, it's typically 23. A haploid, a cell is haploid when it's just n or 1n, and that's just one set of chromosomes, which is basically one of each type of chromosome, no pairs. Now, when a cell is told to be diploid, what happens is that there is two sets of chromosomes, or 2n, and two of each type forming homologous pairs like this example here. Now, when a cell is referred to as polyploid, it means that it is anything more than two sets, so like 3N, 4N, 5, and so on. Now, chromosomes can basically come in distinct types. Basically, with humans, they have two sets of chromosomes where there is 46 in total. Every somatic cell basically has a set of chromosomes in their gametes. In their so the humans in their somatic cells have two sets of chromosomes, but then in the gametes it only has one set, which is 23. Now they are in pairs, like we were talking about, where there's 22 pairs of autosomes like this, and one pair of sex chromosomes. The sex chromosomes are the ones that determine the sex of the individual itself, and the others are called autosomes. So now some terminology with chromosomes is to just keep in mind is that we have unreplicated chromosomes that look like this, and then there are replicated ones from each parent. And then there are homologous chromosomes, which are basically from different parents. Sister chromatids are from the same parent, but it's just replicated and combined. And then the centromere is the part where it gets combined with the sister chromatids. And then with chromosomal replication is basically the replicated chromosomes like this. Now, one thing about chromosomes is that they change during the cell cycle itself, where eukaryotic chromosomes consist of DNA associated histone proteins. And in this case, when the DNA and the protein are combined together like that, they are called chromatin. And when it's relaxed and like thread like this, it's called euchromatin. It's basically unzipped. Now, when it's like zipped up like this, it's called heterochromatin. It's basically condensed before the process of mitosis occurring, like a zipped file. Now, one thing about mitosis is that it has 5 subphases to them. And these include prophase, prometaphase, metaphase, anaphase, and telophase. In the first stage itself, prophase, this is where the chromosomes start to condense down and start looking like the chromosomal shape. The mitotic spindle forms the microtubule organizing center, which is basically called a centrosome in animals and contains a pair of centrioles. This spindle is basically made of microtubules that basically are called spindle fibers themselves. Now, this is what mitosis looks like in like a plant cell, where it kind of goes into prophase here. There are no centrioles in plants, but a mitotic spindle is formed from the microtubules itself, but it's not um, uh, properly organized or understood how these are actually organized. Now, the next step of this is called prometaphase, where the nuclear envelope is breaking down and the nucleolus disappears. The kinetochore microtubules attach to one of the sister chromatids to each of the different chromosomes. So one attaches here, one attaches here, and starts to kind of allow the attachment to occur at the centromere region with the kinetochore itself. So the attachment of the kinetochore basically kind of starts with like the mitotic spindle, and it starts off and it attaches to each sister chromatid at this region, more like here, where the sister chromatids are connected. And slowly, when it goes into anaphase, it breaks off and gets pulled away from each other. So when it occurs with metaphase, what happens is that this mitotic spindle is completed, and the kinetic core towards pulls each chromosome in opposite directions, causing the uh, chromosomes to line up in the middle of the cell like this. Like, so meta meaning middle. It basically forms an imaginary plane in the middle of the cell where it's called metaphase plate, and it just lines up in the middle. And what happens in the next phase during anaphase, what happens is the centromeres themselves end up splitting, and it gets pulled apart towards the opposite ends of the poles. So basically, those end up splitting into two unreplicated sets. And then, as soon as they are no longer attached, they become the sister chromatids become chromosomes and gets separated as they move towards the separating poles. Now, how do these chromosomes actually move during mitosis? And this is typically through the process of the kinetic core microtubules shortening, because the tubulin subunits itself are lost from the ends. So when we look at this, is that when it gets pulled away, it starts getting removed. It's like detach, move, reattach, and just kind of goes on towards the poles themselves. So it moves towards the pole itself as it releases. Then we have the telophase stage, where the new nuclear envelope begins to form around each of the chromosomes, and the mitotic spindle itself disintegrates. The chromosomes end up relaxing a bit more, and mitosis ends up complete with when two independent nuclei have formed in that process. Now, there is one mechanism afterwards of after nuclear division. There's also the cytoplasmic division, where it's called cytokinesis in plants. It's basically the vesicles from the Golgi being transported to the middle and dividing the cell where they fuse to basically form that cell plate. Within animals, fungi, and slime mold, what happens is a ring of actin basically comes in with myosin filaments contracting inward to form a cleavage furrow, and just tries to bring it apart. Now, one thing about bacteria is they do it similar to animal cytokinesis. It's basically called uh, cell division by fission. This is basically when the DNA is copied, the copies end up separating, forming like rings, and then the ring itself comes around, draws in that membrane, and then splits it apart into two separate cells. Now, one thing about all of this is that there are signals when to divide and when not to divide. And one of the main cell cycle control proteins, it's is called mitosis promoting factor, and this is there to kind of induce mitosis in the different eukaryotes, and its levels are very tightly regulated. If they're not tightly regulated, it ends up with various kind of diseases where it's affecting like proper development or like cancerous kind of scenarios. Now, this MPF, or the mitosis promoting factor, it's basically composed of two distinct subunits, where it's going from the protein kinase and the cyclin subunit. And in this case, there are different components of it throughout the different phases of the cell cycle. There are three different checkpoints. One is the G1 checkpoint within interphase. So there are a couple of stages in interphase that is allowed, and these are include G1 as a synthesis phase, and then the second growth phase. And there are a couple of different checkpoints where to make sure that the cells are growing properly, they have the right nutrients and all that to kind of get through the different checkpoints. So the growth one phase, or the first gap phase, what happens is they want to make sure that there's the right cell size, if there's sufficient amount of nutrients, any social signals that are telling you to divide, and the DNA is properly there with no damage. Then it goes into the S phase, which basically replicates the DNA, where we will talk about that in a couple of lectures from now. And what we see here is we see that within the growth phase 2, what happens in that checkpoint is if they have been uh, correctly replicated and the DNA is undamaged and the MPF that we talked about is present. And then once it passes that, it makes sure that it goes into the M phase checkpoints, where they check the chromosomes are attached to the spindle apparatus during metaphase. And what happens is they want to make sure that it's properly segregated, and the mitosis promoting factor is not there anymore. So now all of this kind of is regulated by cyclins. These are components that are there to kind of just make sure that it's being regulated correctly. If so, the active MPF concentration increases during interphase and peaks in M phase before decreasing again. And then the MPF CDK levels, they end up constant throughout the cell cycle. So it kind of goes consistent throughout the cell cycle. And then the cyclin levels increase during interphase, peaking at mitosis, and then comes back down. So the concentrations are high, MPF is more active. If we have more target proteins, and end up phosphorylated to initiate the mitosis cycle itself. Now this might be another big question that we need to think about. What happens, or what actually determines the amount of times that a cell divides? Typically, there is a preset number of cell divisions that a normal cell type can go through before they become quiescent, or functioning in a way that it's not dividing eventually, basically senescent and end up dying. And this is referred to as the Hayflick limit. So once they kind of reach that limit, they stop dividing, but they still do their functions. Now, when it comes to like stem cells, what happens is that these stem cells end up undergoing many more divisions, but are still responsive to like growth signals around it. And then with cancer cells, what happens is that they end up with the unlimited ability to end up dividing, and even in the absence of growth signals. So like it just goes on and on with no limit whatsoever. Now, one of the major checkpoints to avoid any like issues with like cancer and all of those, it's basically checkpoint protein that is called P53. This protein basically detects DNA damage, either pauses the cell cycle in G1 and go to G0, which is the exit kinaside, and basically initiates apoptosis, which is basically another word to talk about programmed cell death. And P53 here is basically a tumor suppressor protein, and the absence of the P53 protein can lead to uncontrolled cell division and basically leads to many types of cancers. And if both copies of P53 gets knocked out, it can lead to many different types of cancers by any type of different environmental uh, uh, triggers or a genetic related issues and all that. With that, we will pick up on the meiosis, the other type of cell division in the next video.